Hello everyone, my name is Fayaz Khan Sahil and today I'm gonna teach you guys about cells. So as you can see, this is our learning objective that is directly taken from the syllabus of O levels. Let's start with the definition of cell. So what is a cell? A cell is a basic unit of life. It's like a tiny building block that make up all living things, from bacteria to human. Cell could be singular or multicellular. Singular cells such as bacteria, viruses, although that is contradictory. Some scientists believe virus is a living thing or other doesn't believe virus is a living thing because they can only replicate inside a host cell. Okay, let's move on to the diagram of an animal cell. So you can see a detailed diagram of an animal cell. You can see the cell membrane. As you can see the lysosome are vesicle, so they basically contain enzyme, suppose protease, in that sort of. So let me change the, okay, I guess now it will write more smoothly. Protease. Okay, so you can see the cytoplasm, you can see the centrosome, you can see the rough endoplasmic reticulum, you can see the smooth endoplasmic reticulum, you can see ribosome, Golgi body, mitochondria. We'll learn all about it. Now let's move on to the functions. Nucleus. So inside the cell you can always see there is a nucleus, right? For example, suppose this is an animal cell. You will always see under a light microscope, you will always see a nucleus and a dense region. So what is a nucleus? Nucleus contains genetic material which controls the activities of cell. So what does nucleus actually generated? You will learn that thing in details, but for your kind of information, when the DNA, suppose this is an DNA, when it get translated, it produce a mRNA that is translated into protein. And how does this control the cell? How does manufacture of protein control the cell? That is a very interesting question. If you can see, if you can just imagine, all the enzymes are made up of protein, and protein control all the chemical reaction inside the cell, most of the chemical reaction inside the cell. So as nucleus produce this enzyme, this is how they control the chemical reaction inside the cell. And this is my first video, so bear with me. I'm a little bit nervous to talk in front of you guys. I don't know where you guys are from. Please write that down in the comment section, and I will try to post and help you guys through this journey. I have given my A levels in 2021, so it's not that far. I mean, it's far. I'm getting older. So let's look at the function of cytoplasm. Cytoplasm is a gel-like substance composed of water and cell solute. It supports the internal cell structure, and it is the site of many chemical reaction. So the question should be, why is the site of chemical reactions, right? This is because cytoplasm are made up of 70 to 80% of water. Okay, and water is where the most of the chemical reaction actually take place, right? Now let's look at cell membrane. Holds the cell together. What is the function of cell membrane? The function should be that is holding the cell together, right? If we look up at the image, you can see it is holding the whole cell and its organelles together, right? Suppose it got broken, right? Suppose you break it up, right? You have a superpower, you break it up. So what will happen? All of these organelles will flow out of the cells, right? The cell will die, right? So the function of cell membrane: hold the cell together, separating inside of the cell from the outside, control which substance leaves and enter the cells. Cell membrane is quite fascinating. It always fascinated me in biology. Let me show you guys the diagram of cell membrane. And my handwriting isn't that good, so bear with me. Look, cell membrane is actually made up of phospholipid bilayer. Okay, I'm showing you guys a simple example, but cell membrane are usually far more complex than this one that I'm drawing. Okay. By the way, how you guys preparation for O levels and A levels? Are you guys panicking or something? I remember in my time, I didn't panic at A levels, but I did panic at O levels because I was so scared to give the examination. So I hope all of you guys are doing really well. So we are done with drawing the structures. So as you can see, this is known as phospholipid. So cell membrane is made up of phospholipid, right? And these are like protein channels. For now you just learn about these are protein channels because I don't want to complicate the things for O levels. So as you can see, the passage is very narrow, so it allows selective substance to pass through it, as it explained over here. It controls which substance could leave and enter the cell. For example, through this passage you can say sodium ion Na plus could pass through it very easily, right? But however, suppose this is a protein, right? Suppose this is a protein. Can protein pass through it? No, it cannot pass through it. The passage is too narrow for protein to pass through it. Okay, did you guys get it? Now let's rub this thing off. Okay, now you can see the next we have is a ribosome. Okay, ribosome are found in cytoplasm, where these are the site of protein synthesis. So if you look at endoplasmic reticulum, if you look at rough endoplasmic reticulum, let me rub this thing off. If you look at rough endoplasmic reticulum, you can see the small dots, right? This dot is known as ribosome. They are found in both attached to the rough endoplasmic reticulum, and also they are found in free body, right? They are moving around the cell in the free body. Now let's look at another type of cell. You are very used to it. So interestingly, some organelles are found in plant cell, however it isn't found in animal cell. Okay, for example, cytoskeleton, perioxisome, ribosome, vacuole. So you can see some organelles are only found in, no, these are not the organelle that is only found in, you can find temporary vacuoles in plant cell too, in animal cell too. Sorry, I'm stuttering a bit, so bear with me. By the way, I'm currently doing microbiology and genetic engineering, so I will try to guide you guys through this O levels to A levels journey. Just bear with me. I'm a little bit shy, and it's a lot for me to open up a camera. Some of you can understand my feeling. Some of you are very introverted. You can understand what I'm trying to say. So what are the organelles that can be found in plant cell but not in animal cell? Let's look at it. Cell wall, chloroplast, and permanent vacuole. So cell wall. What is cell wall made up of? This is a very interesting question. Cell wall, what's your opinion? What is cell wall made up of? Cell wall is made up of cellulose, okay? So cellulose is nothing, just made up of glucose, right? Beta glucose. You don't have to remember this thing. And chloroplast, what about chloroplast? This is where photosynthesis actually take place. Okay? This is a site of photosynthesis, right? And permanent vacuole. They have a permanent vacuole which store various nutrient and salt, right? Let's look at their function. Cell wall made up of cellulose give extra support to the plant. Why does we need it? Extra support. You will understand in the chapter osmosis. That is the cell wall is very important. Now let's look at the function of chloroplast. Chloroplasts contain green chlorophyll pigment that absorb light energy during photosynthesis, right? And what is the function of permanent vacuole? Permanent vacuole contains cell sap, which is a solution dissolved in sugar and iron. It is used for storage and support of a cell structure. If you guys should take notes, taking notes is very important for O levels because you cannot read the whole book before paper 2. Now let's look at one of the most interesting thing for me. It's a bacteria because I'm gonna be a microbiologist. So for me, bacteria is something I deal with every single day. So what do you know about bacteria? Let's look at few of the structure that is similar, which is cell wall, but it is not made up of cellulose. You will learn it later. We have ribosome, which is common in both, which is common in three of them: animal, plant, and bacterial cell. We have cytoplasm. We have bacterium flagellum. This structure you might be not familiar with. Flagellum is not a permanent structure, okay? What I meant is that it is not found in all of the bacteria. So the use of flagellum is for movement. So the bacteria which have flagellum can move through the space. It could be water, or it could be your delicious juice that you guys eat. So before I started microbiology, I wasn't very sensitive about the food I'm consuming. But after I started to research, oh my god, normal food substances contain so many bacteria, and it's sometimes get over my mind. Now I'm very, very careful what I consume. So we have DNA, plasma membrane, but however, interestingly, you are seeing the DNA doesn't surrounded by an envelope. It isn't. It is free living, right? It is floating. It doesn't surrounded by a nuclear envelope. That is one of the major differences between them. So let's look at what is a bacteria. A bacteria are unicellular organism with a simple structure. They are much smaller than plant or animal cell. The bacteria are very small to see. You need specialized microscope to see a bacteria. So they are smaller than both plant and animal cell, surrounded by cell wall, but it is not made up of cellulose, right? Like plant cell, okay? Let's look at some of the function of it. Cell wall, it is made up of peptidoglycan. It is not made up of cellulose. So we have learned the function of cell wall is to provide structural support and protection. It maintains the shape of the cell, prevent the cell from blasting due to osmosis. For example, I don't know if you guys are familiar with the topic of osmosis, but I'm guessing you guys are. For example, suppose this is an animal cell. This is an animal cell, okay? Let's draw a nucleus. So what happened, it doesn't have a cell wall. Okay, so if water enter this thing, enter the cell, if continuously water entered the cell will eventually blast. This is because of the pressure that is provided by the water molecule that is inside the cell. This is known as target pressure. Okay, so if you have a cell wall, suppose if you have a cell wall over here, then it cannot be a animal cell, it could be a plant cell or bacterial cell. As you have a nucleus, so it is a plant cell. So it provides structural support. For example, if this water molecules start to give pressure, the cell wall will give equal amount of pressure, or more pressure or less pressure. So it helps to maintain the structure and provide protection to the cell. It also helps to maintain the shape of the cell, right? This because of this cell wall, plant cell can maintain its shape. For example, the most common diagram you can see is this, right? So it can help to maintain the shape. It prevents the cell from blasting due to osmosis, as we have explained before. Now let's look at the structure of cell membrane. It's just like any other cell membrane that I explained to you guys, right? It is made up of phospholipid bilayer. Why is it called phospholipid bilayer? The simple answer is that you can see it is made up of two phospholipid layer, right? That is why it is known as phospholipid bilayer. Bi means two, right? So what is the function of cell membrane? We have learned literally few minutes ago. So the function of cell membrane is to control the movement of substance, right? For example, there is a protein channel. There is a protein channel. So not all substance can move through it. Only specific substance can move through it, right? It involves in respiration and transport in some bacteria. Okay, now let's look at the cytoplasm. Cytoplasm is jelly like fluid filling in the cell. It is as we all know, cytoplasm is a site of metabolic activity or metabolic reaction. It contains enzyme and other molecule needed for life and hold the ribosome and DNA, right? That is a function of cytoplasm, as I previously mentioned. Please take notes. These notes are well structured. I think it will help you guys a lot in the future. Now let's look at the circular DNA. Interestingly, it is made up of single circular DNA molecule floating freely in the cytoplasm. It carries the genetic information for the cell and control the cell growth, reproduction and activities. What is the function of plasmid? Plasmid are small circular DNA. So interestingly, I will explain you guys this one. I think this is very interesting. So you can see these are like bacterial main DNA. Okay, main DNA. Okay, but we have a circular substance which is also made up of DNA. We call it plasmid. What does plasmid hold? This plasmid can be transferred from bacteria to bacteria. Okay, it can be transferred from hair to hair when it's cloned, right? So what is the function of plasmid? It contains extra gene. For example, it provide antibiotic resistance and enzyme to digest unusual substance. For example, these genes can be transferred from this to this. That's why nowadays we have so many antibiotic resistance bacteria. Now let's look at the ribosome. We have learned about this dot-like structure in the eukaryotic cell. So this dot-like structure is known as ribosome, and their function is for protein synthesis. They have only one function, which is used for protein synthesis. They are only used for protein synthesis. And why does we need protein? Protein are needed for cellular function. Like all the enzymes are made up of protein. So you can say without protein our cell cannot function. We have other optional structures such as flagella. I have told you it is present in some bacteria where isn't present in some other bacteria. It is a tail-like structure used for movement. Villi, which is a short projection that help the attachment to surface to other cells. You will learn that thing in great details. If I try to explain you guys, you wouldn't learn that thing properly. Now we have capsule. So sticky outer coating that protect the cell from dying out and help to evade immune system. Okay, there are some bacteria which are super villains. For example, I cannot say Thanos is a villain. I don't know. I don't know. Some of you guys think Thanos is a fully villain. I don't know if he is a fully villain or not, because he was only doing that thing to save the humanity in his perspective by killing a lot of people. But I don't know if you are if you are Marvel fan, please let something down in the comment section. Just write it down if Thanos is a villain or not in your perspective. So, sorry, excuse me. So some bacteria are actually villains. For example, suppose this is a bacteria, okay? This is a cell membrane. Then we have a cell wall, okay? However, they have another capsule to protect them up. So you can see, you cannot invade those villains, right? They are protected by this slippery capsule. By this capsule. For me, this is actually very interesting. Some of you guys gonna be a scientist, and your main job would be protect against this resistance bacteria, right? Now let's look at the microscopic examination of animal cell and plant cells, right? Let's look at the diagram of light microscope. We have eyepiece, we have condenser. The main function of condenser is to condense light. We have a light source, we have objective lens, okay? These are used for adjustment. These are like carrying handle which hold the objective lens and eyepiece. So you can see the overall structure of a light microscope. Now let's look at what is a light microscope. A light microscope is a tool that help us see very small thing like cell using light and lenses. First, light microscope shine through the object on a slide, then the light goes through the lens and make the object bigger. The objective lens closer to the slide and the eyepiece lens which you look through work together and magnify the image. You can put the slide on the stage and use to focus knob to make sure the image is clear. So now you are seeing what the hell this guys is explaining, right? Like this sounds so complicated, but this is very easy. Let me explain you guys. For example, this is eye source, light source I meant. So this is your specimen is over there, for example, okay? So what will happen is that light will pass through this condenser will condense the light, and then it will passes through the objective lens, okay? Objective lens, this objective lens, suppose it's 10x, for example 10x. For example, your specimen size is this, it will magnify it 10x, okay? This, after magnification, these light go through the eyepiece. Eyepiece also have magnification. So your image is already magnified 10 times, right? If eyepiece lens is also 10 times, right, if light passes through this, so the resultant image will be 100x magnification. As you can see, the magnification in done in two way, for first in the objective lens, then in the eyepiece. Okay, so you cannot just see a bacteria or see a animal cell or plant cell just sliding under the microscope. Okay, first you need to stain the cell. So for animal cell we use methylene blue. It binds with the DNA and stain the nucleus dark blue. For example, these, oh no, I cannot use this color. For example, this is an animal cell, this is a nucleus, this is a DNA. Okay, I'm just showing you a shortcut. Okay, for example, what methylene blue will do, suppose this is a methylene blue. So what does methylene blue will do? This methylene blue will travel to the DNA, travel to the nucleus and to the DNA and bind with it, making the nucleus blue. Did you got it? Now let's look at the procedure of staining. First, place the cell sample on a microscopic slide. For example, this is a microscopic slide. You place that at the middle. This is your cell sample. I should mark it with red. This sounds look nice. Then what we'll do, you drop a stain. Your stain is blue, right? You drop a stain. Okay, these are your stain. This is the sample. This is the stain. This is a microscopic slide. So you can see over here, you add the methylene blue over here, it will stain the DNA. And now we'll put a cover slip. Cover slip is usually put it above the sample. Okay, suppose this is a sample, this is a microscopic slide, it will put above it. Okay, now you put a cover slip so that no dust can enter through the samples. Okay, now you will observe under the microscope. Okay, now we'll observe under the microscope. What image will you see under the microscope? If you see under the light microscope, you can see a nucleus. You can see nucleus. You can see mitochondria. Let's zoom it out a bit. I think we can see mitochondria over here. This is a mitochondria. This is mitochondria. We can see cell membrane. And cytoplasm. These are like cytoplasm, right? So my question is, can you see ribosome under the light microscope? The simple answer is no, you cannot see ribosome under light microscope. Okay, now let's look plant cell under light microscope. Under a light microscope, you can see the main structural component of plant cell, including cell wall. You can see the cell wall is visible. You can see the cell wall is visible. You can see the nucleus is visible. You can see the cell membrane and chloroplast is also visible. But this image is not fully clear. Okay. So what is the staining method for plant cells? The staining method including iodine solution. Now we do not use methylene blue, we use iodine solution. Okay, so what does iodine solution do? Suppose this is a plant cell. Okay, this is the plant cell. This is the cell wall. Cell wall isn't that big, but I drew it. And this is a nucleus. Okay, So what we'll do is that it will stain the it will stain the cell wall and the nucleus. It won't stain the cell membrane, but it will stain this one. Let's learn about electron microscope. Okay, electron microscope is a powerful microscope that uses beam of electron instead of light to see very details of tiny object like inside of cell or viruses. It can magnify object much more than light microscope. So the capability of electron microscope is much larger than light microscope. So why do we need that larger magnification, right? Your question should be that we need this larger magnification to see ribosome, mitochondria in more details, and even DNA strand. The strand of the DNA could be seen with this electron microscope, right? So however, you just cannot just look at the eyepiece and look at the image. It doesn't work like that. Instead of light, it uses electron beams. So you can see a X-ray image of the slide that you have taken. There are two type of electron microscope, TEM and SEM. TEM is transmission electron microscope, which is used to see inside the cell. For example, you wanna see inside the cell. Whereas there is scanning microscope to see the surface of the cell. Okay. So for today we are done with the lecture. Please do subscribe to my channel, and I promise I will make better and better video. With each video it will get improved. And please take the notes. The notes, if you memorize it, it will really help in your examination. In the next class we'll do 1.2 unit 1.2 of cells, which is a very small one. Bye bye. See you next time.